Hey, ladies and gentlemen, this chapter we are talking about cellular reproduction, also known as mitosis or meiosis. This is how cells make baby cells. That's going to be later notes. Today we are going to be focusing on the life cycle from birth through growth to reproduction and death. That's right, death and reproduction are together because cells are asexual, meaning they don't need a partner. They're just going to become the baby cell. So technically, this cell is dead when it becomes the two baby cells. Before we get into the notes, let's look at a broad overview of the entire thing. So you're not writing anything down yet, just watching this. We are talking about the life cycle. So this is like time, which is why I have a clock here. From the time that the cell is born all the way through the clock to the time that it reproduces. But we're not going to use it as a clock. All right, we're going to change this up. Still going to have the arrow. And we got four basic times. We call them the G1 phase, S phase, G2 phase, and M phase. So what's going on in these phases? If you look over here, this is the cell, and we're going to see what happens in each phase. So first is G1 or growth phase 1. The cell is simply going to grow. It gets bigger. It also gains some more organelles and all the other stuff that is going to need to make two cells. Everything except DNA, because as we learned in a previous chapter, making DNA is this whole big process. So it gets its own phase we call S phase or synthesis phase. This is what we learned in that previous chapter, DNA replication, where this strand of DNA gets split, the two sides split apart, and then this side becomes this one strand, this side becomes another strand, and we get, look over here, two strands of DNA. That happens in S phase. And then we get G2 phase, second growth phase, or growth phase 2. The cell gets slightly bigger if it's not done growing, but here's the important part. This thing right here is going to become important. That's called a centriole, and we're going to use it in the reproductive part. And we're going to need another one. So in the second growth phase, we make a new one. How do we do that? Last chapter, protein synthesis. We made the proteins to make that. And then our clock continues to M phase, or the mitotic phase, or simply mitosis. And this is where a lot of this weird stuff happens. We're going to have a whole separate set of notes about all the different steps involved in this, but generally, M phase, mitosis, is all about getting these two strands of DNA away from each other. Unfortunately, they're trapped in the nucleus, right? So we need to get that nucleus to dissolve away. And these guys, I didn't animate it, but they're going to shoot out strings, like we said in the vocab, called spindle fibers, and do a tug of war and pull them apart. And then they each get their own nucleus. And of course, the cell splits in two, and we get two tiny baby cells. Each one will go through the entire process again. And that was just the overview. So let's jump into the actual notes. You should have your guided notes out and ready to go. We are talking about the cell cycle. Before we begin, please remember that we have two different kinds of cells, reproductive cells and non-reproductive cells. And M phase, the mitotic phase, is going to be different for each one. So your reproductive cells, gentlemen, your sperm, ladies, your eggs, we refer to them as germline cells or gametes. These are going to be vocab words later on. And these are what we call haploid. Again, another vocab word later on. We didn't get these words yet. Haploid means they only carry one chromosome. Normally, your cells carry two, one from your mom, one from your dad, right? Your dad gave you 23 chromosomes, your mom gave you 23 chromosomes, so that's 23 pairs. When you make your sperm or when you make your eggs, you're going to take one from each pair and put it in your germline cells. Non-reproductive cells, which are every other cell in your body. If it's not sperm or if it's not an egg, it's non-reproductive, and we call all of them somatic cells. That is a vocab word for right now. You do have this vocab word. So liver cells, kidney cells, pancreas cells, brain cells, everything. And these are what we call diploid or diploid. Either pronunciation is fine. It just means you have two chromosomes in these cells, one from mom, one from dad. Every single cell in your body has its own copies. Each one will go through M phase in a different way. We'll have a set of notes for these and a different set of notes for these. But generally speaking, we're just going to talk about the basic life cycle, not going into those differences right now. The cell cycle is the lifespan of one cell. Remember, I showed you a clock. From beginning to end, it is a time frame. And because it's a cycle, it is going to finish off where it started. As I showed you in the overview, we started off with a small cell and it grew and split into two small cells. And then each one is going to go through its own cycle of growing and splitting, growing and splitting. It's going to always end where we started. And what we are focusing on in this chapter is eukaryotic cells. Why is that important? Because eukaryotic cells have a nucleus, prokaryotic cells do not. And we are going to be focusing on what happens to the nucleus in this chapter. So remember, eukaryotic cells have a nucleus and membrane-bound organelles. In order to make two new cells, everything needs to be copied. Both of these baby cells, we call them daughter cells. Both of these daughter cells have to be completely identical to the original parent cell. So here are those words. Parent cell is the original. The daughter cells are the two smaller cells that form when the parent cells divide. So going into the phases of it, you'll notice that this looks very similar to the clock format in my original overview. It's upside down. I had M on top and S on the bottom last time. It's fine. It's a cycle. You can spin it around and it's all good. Right. Remember, we started off with growing. The cell got bigger. We made more organelles. We got more DNA. We got a little extra growing, more construction going on. All of that is just the cell living its life, doing its stuff. And we call that interphase. G1, S, and G2 is all one big section called interphase. When we get into the actual reproduction part, mitotic phase or mitosis or just M phase, this is a very careful procedure with a bunch of steps to make sure we do not mess up the DNA, because that's what it's all about. Mitosis is about separating the DNA in a very, very careful way. We just can't just rip it apart. We have to be very structured. And then afterwards, we have something called cytokinesis, which is when the rest of the cell gets split apart. So mitosis is when we separate the DNA into their own nuclei very, very carefully. And then cytokinesis is when we split the rest of the cell. And then each brand new cell gets its own nucleus with its own DNA. So going into interphase, we have G1 phase, which we saw last time. First, growth phase. We are going to make more cytoplasm, which is what makes the cell bigger. Remember, cytoplasm is all the liquid goop on the inside. And we're going to make some more organelles. So it gets much bigger. S phase, or we call synthesis phase, making new DNA. Remember, guys, your DNA isn't always one big long strand of DNA. We have it chopped up into 46 pieces that we call chromosomes, and those pieces get braided up. So when we look at it in a microscope, it's actually going to look more like this, braids of DNA. After we go through S phase or synthesis phase, DNA replication, whatever you want to call it, we're going to get twice as much of these things. So now we have two yellows, two dark blues, two light blues, two greens. You get it? And oh, that was a mistake on my part. There should be two of those guys, the bluish gray one. Go with it. And eventually we're going to have to split them up, just not yet. And then we got G2 phase, a little bit extra growth, but we're going to make the structures needed for cell division. We call these centrioles, and we're going to worry about that in a future set of notes. So we double these up, and then we're done interphase. That was easy. Yeah, that was just stuff that we talked about this whole year. Growing is just protein synthesis, with the RNA and the ribosomes, all that stuff. S phase was just DNA replication. So everything we learned in the last chapter was interphase. Now we need to get ready for the next step, nuclear division. That's what we call mitosis. We need to separate these brand new copies of DNA. So first thing's going to happen, these centrioles are going to go to opposite sides of the cell, and that nucleus is going to dissolve away. Just bye-bye, because we can't separate them if they're trapped in a bubble. So that just breaks down. Then these centrioles are going to release a special chemical that your DNA hates. So it's going to try to run away from these centrioles, but it's also going to try to run away from these centrioles. So that means they're going to be stuck, lined up in the middle. That's a good thing. And then I didn't animate this, but we talked about it in the last set of notes. These guys are going to release fibers coming out and grabbing each side. And there's going to be a tug of war ripping those chromosomes apart. And they get pulled to opposite sides. And once they are piled up over here, they each get their own nucleus. And take a look. We have yellow and yellow, green and green, red and red, dark blue, light blue, teal, teal. They are exactly the same. And then the final step we call cytokinesis. This is separate from mitosis. Mitosis was all about getting the DNA separate. Cytokinesis is all about splitting the cell in half. Animal cells and plant cells are going to do this differently. We talked about this in vocab as well. Animal cells are going to create something we call a cleavage furrow, where the membrane pinches inward and the DNA is going to go on one side and this DNA is going to go on the other side, and we end up with two cells. Plant cells are going to do it a little bit differently because remember, they have a cell wall. They can't pinch inward like this because that cell wall is made out of, well, basically wood. So try pinching in, it's going to splinter and we're going to destroy the cell. So plants need to be different. Instead of pinching inward from the outside going in, they're going to start constructing from the inside out. And we call this construction a cell plate. It's going to be made out of cellulose, the same stuff as the cell wall. And when it's done growing right down the middle and reaches both ends, it will become a brand new cell wall, and we will have two cells on either side. And that's the life cycle of one cell. It's made, it grows, it reproduces. So one last quick recap. G1, it grows. In S phase, we make more DNA. In G2 phase, a little bit more growth, and we make a new centriole. And then M phase is the complicated one. Centrioles move to opposite sides, the nucleus dissolves, the centrioles pull the copies apart, they each get their own nucleus, and then cytokinesis happens. That is the cell splitting into two smaller cells. That's it, ladies and gentlemen. Please go check Google Classroom. There should be a worksheet waiting for you, and I will see you in class next time. Have a great day.